I did some reorganizing to Canvas, if some of you guys had noticed, the past couple of days. I didn't like the way that it was. It just wasn't working for me. So I changed it. So you'll notice I even put a link and did an assignment link so that I can start grading. And so I want to go to that right now, one of the assignments that we're working on for this module. And so this links to that page, which you guys are editing, right? You're adding your name, and do your 3-letter initials, because in case there's another person that has the same 2 as you, right? You might want to add your 3rd letter just in case, so that when I grade, you'll get your points. But basically, if your name is on the list, you should get your points. But the important thing is if you don't get something right, I need to let you know. So we have 3 spots left. We need to fill those today, right? So somebody please jump on. The last person to fill the last spot in the top table, please send me a quick email. Let me know that it's done so that I can check this table and make sure that it's correct before we move on to the next table, right? Which we need to get done. Mine's still on too. Turn your cell phones off. Okay, any questions on this? Okay, there's also 35 people in this class, and I only did 34 spots, so I'll be adding a spot at the bottom. Add one more chemical down here. This is only the beginning of this assignment on chemicals to clean. The next part is going to be a discussion board, and it's going to be fun, right? I've done this with my other classes, and they like this. The only problem is part of that discussion board, people weren't able to answer the questions because we didn't go over this material first. And so I felt that maybe getting you guys to compile the information ahead of time would help you in the discussion board. And what the discussion board is you're going to basically look in your home for something that you clean with and see if it actually is disinfecting and to what level, right? So it's kind of fun activity, right? And I bet most of you guys, one of the parts is to actually read the entire label to what you're cleaning with. And I bet a whole bunch of you guys, right, haven't been following the instructions, haven't read the label, right, to the things that you're using to clean your house. So it'll be a good life skill too that we'll learn on this activity. But we need to get this table done, right? So most importantly, we need to get this table done today. So please, 3 more people, right? So you remember how you do this is you click edit. You add your name to the list, right? And then for this table, it's just yes or no whether it destroys, right? So if it's considered an intermediate level disinfectant, can it destroy enveloped viruses, right? Or no. So yes or no, right? And this information is in your book, right? You're not having to search anyplace else. It's there. Bringing me to one more thing. There's a few people, your license has expired for Connect. Connect is a required element in this course. You need to take care of that ASAP. And then I think there's one person that hasn't even registered. So if you're confused about that or not sure what you need to do, the good news, since you haven't registered, you could do free access for 2 weeks, right? And the test is coming up, so you want to do these assignments, right? Because they're going to help let you know if you know this material before you take a test. So you definitely want to do that. But come see me if you need help. As always, contact me. You guys even have my cell phone number, pseudo cell phone number. So no excuses. And step by step in my day 1 recording, I show you, right, and explain stuff regarding to that. And I put the Connect link just about everywhere, right? So, and once you register the first time, it's a single sign-on. So once you hit Connect, it goes right to Connect. You don't have to sign on to Connect separately. But it is a separate website. I'll open in a separate tab, right? That's what it looks like for me, but this is what it looks like for you guys. And notice all assignments are unlimited, right? I changed the date. One of your classmates was kind enough to remind me that I moved the test, so I needed to move the dates, right? And so by the test, and I just didn't feel like, putting a time there. But you really need to complete those well before you take the test, right, to let you know how you're doing. You can do them an unlimited number of times, which means go ahead, take it with your notes closed, right, with your book closed. See what you actually know. And then you can take it again with your book open, right, make notes for yourself. Plus, I think I even have it set where you can print the questions too if you want. You can, I would suggest if you're one of those ones, write them maybe. But don't memorize these questions. Did we go over this last time where sometimes they go into a little bit more detail than I go into in my class? Okay, so I can't think of a specific example. I'll find one for next time. But if they go into a detail that I didn't go into in my lecture, you don't need to know it. Does that make sense to you guys? So remember, what's your study guide? The objectives. What do I go over in my lectures? The objectives, right? That's what you need to know, okay? Not every single little detail in your book, not every single little detail in the Connect assignments, because I can't edit out, right? But I've still picked these to match our objectives as best as I could. But I didn't write these questions or set these up. Does it make sense to you guys? So the other question I get is, why do we have to do this? Guys, do this because this is the compromise that we've made at this point to not having standardized tests in science for final exams. Okay? So do this because this is a way for us to track across the school how students are doing, right, in these subject matters, as opposed to doing standardized final exams. Okay? So if you don't do this, that's what will become of it, right? If you complain you don't do this, right, you get standardized final exams, departmental final exams in science. I don't think any of us want that. Right? So this is the compromise, just FYI, that's why we do this. Okay? But I actually spend the time and pick questions for you guys. Right? I don't just randomly pick them. So they are worth your time and effort. So in the reorganization of the module, just so that we don't have this huge long page, what I've done is the recorded lectures, I've made a page for that, right? And I've dated each one and the topic. So after today, I'll add the one for today right here. Okay? And then, let me go back to the module. Where is it? So, and then the PowerPoints and the objectives too, I put all on one page. And then again, I put a quick Connect link here to remind you guys about Connect. This was my old system that I was doing that was starting to get too messy. But I just left it there just in case anybody still wanted access to it. Because it also has some animation and stuff links there. But I'm going to put that all on one page from now on. So, any questions on any of that? Okay, so I haven't been lecturing you guys. I've just been going over some stuff in Canvas and Connect and stuff like that. So make sure you listen to the recording. Those of you guys that came in late today. I know there was traffic today and fog, so I was like, where is everybody? So I guess it's good that I needed to do a little shop talk with you guys. So then I'm going to lecture. Thirsty this one. Anybody? I hope you guys all had a wonderful Valentine's Day. I know I did. I found out I don't have breast cancer. That's always a best Valentine's Day gift. So I get to stay with you guys and I have to freak out about leaving you. So I'm very happy to be here today. And my husband treated me very well. So I hope you guys treated your loved ones well, right? You don't need a husband or a boyfriend or a girlfriend or vice versa. Just remember your family loves you, right? Everybody's got a mama and a daddy. So we're going to go to our lecture. And we left off talking about transposable elements, right? And I was giving you guys a real research application on how this is sometimes used. So we're going to talk a little bit about the effect UV light has on DNA. And this is important because all of us are exposed to UV light, right, on a daily basis from the sun. it does damage our DNA. But remember, why is it that we don't have as much mutation as we potentially could? What can our bodies, what can our cells do when DNA is messed up? It can correct it. It can repair it, right? So we have repair mechanisms for UV light damage, and so do microorganisms, some of them more so than others. But when we expose microorganisms to UV light, we do it in very high quantities, right? We have UV lights in our hoods for when we're not working in that biological hood to help keep that surface clean. Because as you guys will learn, and if you haven't already in the control chapter, UV light can be used to control the growth of microorganisms because it can kill them, because it damages their DNA, right? And we're going to look at that specific damage in a second. So tanning beds. What is that? It's UV light. Is that a good idea? Not really. Not really. Because you're overexposing your body to large amounts at one time. So you're creating a lot of DNA damage for your body to repair. If it doesn't repair that properly, what could you potentially end up with? Skin cancer, right? Spray tan, right? There are other options. I'm sure the tanning people hate me, but you know, it's not a good idea. It's not a good idea, right? Because this is what you're doing. In your DNA, in your code, if you have two T's next to each other, two thymines, and they're hit with UV light, they tend to covalently bond with each other. Anyone know what that means? How do molecules bond with each other? How do the two interact? What are they sharing? Electrons. And in this case, they're sharing electrons, right? That's a very strong bond. Unlike the bond that holds the two strands together, what's the bonding that holds the two strands together? It's hydrogen bond. Yeah, she's like, oh, I remembered it. Hydrogen bonding, right? That's not as strong as a bond as covalent. The problem with covalent bonding here between the two thymines is, one, that's not what's supposed to be happening. Two, it distorts the DNA. Do you see this buckle here in the backbone and that sugar phosphate and in here? Remember how we talked about how important the structure of that spiral staircase is? We get a bump in the banister here. Does that create a problem? Yeah, because your enzymes that need to slide down that banister and read that code can't do it. You got to repair it. You got to fix it. Otherwise, whatever gene that's there right now, it can't be used. DNA, you can't copy it, right? You can't have RNA polymerase read it to make messenger RNA. So if you need that segment of DNA right now, it's useless to you. It cannot be read unless it gets fixed. Like I said, we do have repair mechanisms, right? But you don't want to overwork the system. When we expose microorganisms to this, we bombard them with more than they can handle, more than they can repair. And we can slow their growth, we can even stop their growth, we can even kill them. Again, it all boils down to we're distorting their DNA. That's what the UV light does. It affects these thymines, causes them to form these dimers, two bonded together, and they're sharing electrons. It's a very strong bond. Make sense? Okay, so UV light, not so good. So then the last things that we're going to talk about are all linked in that they all are mechanisms of horizontal or lateral gene transfer. So this is all genetic information, DNA, going from one bacteria cell to another. And we're not talking about mom to baby like mutation, right? When that mutation happens in that vertical transfer, we're talking about like cousin to cousin. So did I give you guys the example of E. coli and the Shiga toxin already? Right? So imagine that. Shigella has this ability in its DNA to make this really potent toxin that literally kills your epithelial cells in your intestine. There's a strain of E. coli out there that has this exact same toxin. How did it get it? It probably got it from its cousin, Shigella. How did it get there? Probably one of these three mechanisms that we're going to talk about. It acquired it from its cousin. So the three mechanisms, and sometimes this one's just shortened to transformation, but transformation can be done with other things. So sometimes we specify that it's DNA-mediated. It's the DNA that's causing this transformation. So again, a play on words, right? Telling ourselves little stories, because if you look, the next one, transduction, looks very similar to transformation, right? They both start with the word trans. So sometimes we get in a little trance, right? We don't recognize one from the other. So for transformation, really focus in on, right, that it's transforming, it's changing, right? Its DNA is changing, therefore it changes. And remember, what's the physical expression of the genotype of the DNA called? Phenotype, right? They're physically changing because the DNA changed, right? Because in the case of that E. coli, he got DNA on how to make a toxin. So he could physically make that toxin and make your life a living hell, right? If you had E. coli 0157, you wouldn't be in class right now. You'd be at the hospital. So for me, I always, tell myself little stories, right? Transformation, Transformers, right? Which they're back now, right? They were very popular when I was a kid, right? But Transformers are back. My son is convinced there's a Camaro, right? Have y'all seen it? Someone in New Orleans has a Camaro like Bumblebee. My son is convinced it's going to turn into Bumblebee. Keep trying to explain to him that that's make-believe, right? That that's not going to really happen. A car cannot turn into a robot. But who knows? Someday that could be possible, right? I went to a seminar yesterday here at Delgado, and they're engineering neurons, nerve cells in the myelin sheath in these little matrices. They call them chips. And so they're able to study the effect of drugs directly on nerves, human nerve cells, in like a little petri dish. They're already doing it with like liver cells and kidney, right? Like where they like have organs in petri dishes so they can run tests on them. It's pretty amazing stuff. So, brand new company here in New Orleans working on getting the final pieces together to start drug trial testing on nerves in a petri dish. Crazy stuff. So remember, if that stuff excites you, we have biotech program here at Delgado now. It's pretty amazing stuff. Lots of startup companies that you could get trained here, associate's degree, and plug right into one of those jobs, playing with organs in a petri dish. How cool is that? So again, think of that transformation. Think of that robot and car transformation, right? How one can turn into another. So literally in the microbiology lab too, the end of the semester, my students will transform E. coli to be able to produce green fluorescent protein. So when we shine UV light on it, which excites the electrons, it fluoresces green, right? This is a natural ability found in jellyfish. So we took that gene and just put it into bacteria, right? So some people get kind of freaked out by that, but I got news for you. If you have diabetic friends, they're very happy for biotechnology because the insulin that they have to rely on is made by bacteria now. Human insulin, right? You don't have to worry about getting it from a pig or a horse and having a reaction, right? It's the exact same stuff that we make, but the bacteria makes it for them. And we give it to them. But that's not it. We haven't stopped. We tweaked it because it's a protein, right? So temperature affects proteins, as we'll find out as we start getting into our control thing, right? Why do we cook our food? Denature proteins in the bacteria and kill them, right? So they have to carry their insulin with a little cool pack, right, to keep that protein stable. But they've been able to engineer now a protein that's temperature stable, where you don't have to carry around the ice pack anymore. So pretty amazing stuff. So, we can put stuff, we can transform bacteria to get them to do new things. The problem with this, and if you remember one of the key reasons, why are we talking about genetics? What's the bad news for us when it comes to change of bacteria? They can become resistant to antibiotics, right? So if they change, or if they get an enzyme from their friend that destroys penicillin, then they become penicillin resistant. Right? So it kind of goes both ways, right? So we can make them do things for us, right, in the case of insulin and things like that. But they can gain things that are beneficial this way, right? So how is it that they transform in this case? They literally pick up pieces of DNA from the environment and they incorporate it into their chromosome or into a plasmid and utilize that information. And again, it's information to make something. So they change. They physically change. They physically transform. So hopefully, right, if you're a Transformer fan, right, that whole bumblebee car to robot helps you visualize, too, how dramatically these bacteria can change with DNA information. The next one is transduction. So what's a duct? What's a duct? It's a vessel, right? Something that things travel through, right? So in the case of transduction, that movement of the information goes through something. Anyone know what it goes through? Who's looked ahead in their stuff? How does the DNA get from one bacteria to another? What does it travel through? A virus. Travels through a virus. So again, focus in on the duct. And remember, that's the vehicle. In this case, it's a virus. Conjugation, most people have heard of, right? This is where they actually join up the two bacteria. And they're connected by a structure. What's that structure? Again, it's a little tube. It's a protein hollow tube. Only made by bacteria. Well, that's not true. But the name. Other organisms can conjugate, right? Paramecium can do it. Spirogyra. Algae can do it. Protozoa. It's the sex papillus, right? So it's that hollow protein tube that one cell makes to hook up with the other cell. So anyone know the nickname typically for conjugation? It's the sex papillus. Conjugation is a lot of times just referred to as bacterial sex, right? Because what sex is an exchange of genetic information from one cell to another. So let's go into the particulars of each one of these. So in order for this gene exchange to happen, you must have a donor, right? Somebody where the DNA is going to come from. That donor DNA has to be accepted by the recipient cell. How it gets there, right? Whether the donor picks it up from the environment in the case of transformation, or a virus transmits it from the donor to the recipient, or the two cells hook up to each other within conjugation. The final step, though, the donor has to actually keep the DNA, right? Because although they have the ability to pick this stuff up, they don't have to keep it, right? If you were walking down the hallway and you saw $1, right? If nobody was around, I'm keeping it and going to the vending machine, y'all, right? But if you saw the person walking in front of you drop it, you might what? Pick it up and give it back to them, right? So it depends on whether it's going to benefit you. That's a moral issue there, right? Whether you would actually snag it, even though you saw the person drop it, right? Bacteria are not really moral, right? It's all about survival. So is it going to benefit me, right? If I use this stuff, is this good stuff, right? And so they kind of have that environmental pressure. Remember that selective pressure, natural selection. That's going to help make that determination for them. So in the case sometimes when we change bacteria in the lab, right, unless we have a selective pressure for them to keep that DNA, in the case of making green fluorescent protein, do you really think that's beneficial to a bacteria to glow green? No, and there's no environmental pressure for them to be like, oh, I have to keep this, right? This is expending energy, right, to make those nucleotides and copy that information. That's a costly undertaking of keeping that plasmid we gave them. But we make them keep it. Because guess what's on that plasmid also? Antibiotic resistance. And we grow them in the presence of the antibiotic. So if they don't keep that plasmid, guess what happens to them? If they're in an antibiotic environment, right, what would antibiotics normally do to them? It would kill them. But I gave them resistance to that antibiotic. In that plasmid is the enzyme to not be killed by that antibiotic. So they have that selective pressure that they got to keep it, right? Because if they kick it out, guess what's going to happen? They're going to die. They got to keep that DNA under those circumstances, right? We created a selective pressure on them. So although they can pick this stuff up one of three ways, they're not truly transformed, transduced, conjugated unless they actually keep it. And they don't always keep it. Make sense? Still with me? All right. I know it's early, huh? Still struggling with that 8 a.m. thing. All right. So you need to know the steps in the process of transformation, DNA-mediated transformation. So the first step is kind of sad. Somebody must die. A bacteria must die. The donor, where the DNA comes from, dies. And that's how the DNA becomes part of the environment. So here's a poor bacterial cell that's died, and its DNA is just ruptured out of itself. So that's actually what you're seeing is DNA. So electron micrograph. What shape is this? Bacteria. It's a rod shape, and the term is bacillus, which is also a genus name, right? That's why a lot of us tend to just say rod-shaped. No confusion there. So, bacteria, right? Walking around, creeping around, flagellating around, runs across, in his world, $1, in this case, ampicillin resistance. He has receptors on his surface that can sense DNA in the environment. But usually, they're only going to do this under stressful conditions, right? When conditions are harsh. And so they're like, oh, okay, this might benefit me. Let me try it out, right? So they'll pick it up. When they pick it up, they're going to degrade one strand and only take in a single strand. And then they have to choose where they're going to stick it. So in this case, there's some nominal gene, right, that doesn't give them ampicillin resistance. It's actually making them sensitive to ampicillin. Okay? So in this case, they replace it. And so then they have to replace the other side, right? Or the other choice is, I don't really want it, I'm going to kick it out. This guy, not transformed. This guy, transformed. And remember, once you get DNA, you give it to all your babies, right? So now this is going to travel vertically, right? She's going to give it to all her babies. All her babies are going to be ampicillin resistant. So the definition of DNA-mediated transformation is the transfer of naked DNA. It's all by itself, right? In the environment from one bacteria to another. Frederick Griffith in 1928, which is 8 years after women got the right to vote. My son is studying that right now. It's so much fun. Susan B. Anthony. Remember her? So back in those days, right, he was trying to develop a vaccine for Streptococcus pneumoniae. And there's two different forms. Yeah, I don't have my picture. Two different forms of Streptococcus pneumoniae. One that's encapsulated, which means the bacteria have a capsule. One where they don't have this capsule. So which one do you think is pathogenic and why? Which strain of Streptococcus pneumoniae? So one's got a capsule, one doesn't. Which one's bad news? The one with the capsule. Why? The capsule is protective, right? And how does it help the bacteria in our bodies? So it's a protective layer. It allows them to, guess what? Attach to us. And remember, it's like they're wearing a disguise. They can hide from our phagocytic cells. Our cells are supposed to gobble up and eat the bacteria when they come in. They don't see it. So hence why he wanted to develop a vaccine. So he found that bacteria that didn't have the capsule, right, he could give it to mice, they didn't get pneumonia, they survived that inoculation. When he gave them the encapsulated strain, the poor mouse died of pneumonia just like people do. So he started playing around with it. So he heat killed the strain of encapsulated and gave it to a mouse, and it survived because obviously he killed the bacteria. So the mouse survived. And then for some reason, he decided to combine living, non-encapsulated cells with dead encapsulated cells. And he put it in the mouse. Guess what happened to the poor mouse? He died. And when he autopsied the mouse, he got back out bacteria with the capsule. How the heck did they figure out how to make a capsule? The ones he put in did not, the living cells he put in did not have a capsule. But what else did he put in? He put the dead cells that make the capsule, right? So how do they know how to make that capsule? Where's that information? In their DNA. He basically gave the other cells the DNA and said, here, have this. And under a stressful condition, right, of being in an organism where they got to fight to survive, and there's bacterial DNA there, they pick it up, and they got lucky. They're like, woohoo, I can make a capsule and hide now. That was really bad news for the mouse, and obviously did not lead to the development of a good vaccine, right? But he was like, well, how did this happen, right? So he discovered this, of course, by accident, right? That bacteria can pick up DNA from the environment and utilize it. They can transform. They can change. So we've been knowing about this for a long time, right? Since about 8 years after the women got the right to vote. And we've been utilizing this in laboratory situations ever since, right? Actually purposely causing transformation to get bacteria to do cool stuff for us, like make insulin. So, right, just a lot of words related to exactly what I just previously said. Competent is a term that you'll hear. And again, only under certain conditions do the bacteria do this. So in this case for them, it's stressful conditions, late log, when they're growing in artificial culture, they're running out of food, right? They're starting to enter into that stationary phase where, the population is going to plateau. They're under stressful conditions. So you guys get stressed too, right? If I go, I'll give you the answers to the test, you'd be like, I'm all about that. I am giving you the answers right now. Under stressful conditions, right, you'd be tempted to do things you wouldn't normally do. So this stressful condition can be caused in laboratory setting. A lot of times we chemically make them competent. There's special lines of cells that are more competent than others that are more apt to take up more DNA, especially when we really want them to take it. Right? We really have to stress them out and convince them. They even use electroporation, where they actually hit it with electricity, right, to get the cells to take up the DNA. But in nature, it's when they're naturally stressed, right? And the most stressful thing for them is lack of food. But that's what competent means, right? This state of being able to take up DNA from the environment. So step by step, right, we got to have a donor cell that died and its DNA became part of the environment. You have the recipient cell that's competent, that's willing to take in the DNA, right? It takes it in single-stranded. It finds a place to put it. If it chooses to put it into its chromosome or into a plasmid, incorporate it into the cell, then it's become transformed. And it'll pass it on to all its progeny. So the bad news for us is if this is an antibiotic-resistant gene, right? We normally don't want bacteria in our environment that we're exposed to have these types of abilities, to be resistant to what we're using to kill them, antibiotics. Everyone okay on transformation? Okay. So then the next is transduction. So again, we've got to have a recipient, and unfortunately in this story, the donor dies. The DNA is going to come from somewhere, right? The donor. In this case, he's going to die. What's going to kill him? Or her? It. A bacterial virus. Bacterial viruses are typically referred to as bacterial phage, or just phage for short. These guys are pretty cool. They'll attach to the surface of a bacteria. They'll inject their nucleic acid inside to the bacteria and any enzymes they have in their protein coat. Why don't you think the whole virus enters? What stops the whole virus from getting into that cell? Unlike our cells, pretty much the whole virus gets into our cells. But the bacteria, it's a little bit harder. Why can't they get in? What's blocking them? Why can't the whole virus get in? What do most bacteria have? Why is it holding that shape? Cell wall. Yeah. So they inject their nucleic acids, right, and their enzymes in. And they take over that cell. In that process of stopping that cell from being itself, they'll digest the cell's chromosome, right? They have enzymes that will destroy that cell's chromosome. So they're going to break it up into a whole bunch of pieces. And then they're going to take over that cell. That cell now becomes a virus-making factory. It's going to make copies of its nucleic acid, copies of its protein for its protein coat. In that process, sometimes instead of the DNA for the virus, it accidentally picks up bacterial DNA from the bacterial cell it's infected. When that happens, That virus now is basically just a vehicle, just a duct. It's going to be moving DNA from one bacterial cell to another. When this injects its nucleic acid, it's not going to cause a viral infection. It's not viral DNA. It's bacterial. So this virus just basically ended its life cycle because it screwed up. Didn't pick up the right stuff. And just gave a present to this recipient cell. Here's some DNA from your neighbor on a silver platter. I've injected it into you. And again, if they don't degrade it, right, if they decide to try it out, then they'll become transduced. That virus is done, right? It wasn't really a virus, it was just a vehicle, a duct. This happens even for other cells. They found that this is probably one of the major ways that our algae in our oceans acquire different pigments from each other. It's through viral infection, where a virus picks up the DNA from one algal cell and transduces it, transfers it to another algal cell, and it picks up that information. Kind of interesting. So virus infection could not necessarily be all that bad, right? It could be a present. But in this case, it's not truly a viral infection, right? Because again, the virus didn't transmit any of the stuff to make viruses, just bacterial DNA. Or in the case of the algae, just algal DNA, and not viral. So it's a goof on the virus's part, but a benefit for the potential recipient, potentially. So generalized means any gene of the donor can be transferred. There's actually even specialized transfer where only specific genes can be picked up and transferred. We're not going to get into the specifics of that for this course. We're going to keep it basic. But just know that there's different degrees of transduction that can happen. What can actually be moved from one bacterial cell to another by a virus. In this case, a bacterial virus. So, again, poor donor died. It was infected by a virus. The virus picked up the donor's DNA and it transferred it to the recipient. If the recipient incorporates it and utilizes it, it's began transduced. So I've said the word plasmid before, right? I think we may have even defined it before. Right? Anyone remember what a plasmid is? Right. So it's DNA that can be used by a cell, but it's not necessary for survival. It's extra DNA. It's not part of the chromosome. It's usually circular. It's much smaller. Right? So the difference is much smaller than the chromosome. It's still DNA, though. But it codes for non-essential information, which means they don't absolutely have to have it normally to live, unless you do something mean like we do in the lab and grow it in the presence of antibiotics. So it has to keep its antibiotic-resistant plasmid. Frequently, and this is where it's really beneficial to scientists, when a cell copies its chromosome, it's usually because it's getting ready to divide. For some reason, they'll keep extra copies of their plasmids, though. And actually, plasmids are usually considered high copy number or low copy number. How many copies will they keep inside their cell? So scientists like myself have used them to literally just copy big pieces of DNA, big plasmids, right? Because they'll make a whole bunch of copies for us, and we can just lyse the cell and steal all the copies. They're really cheap copy machines, bacteria. They're great. And pretty effective at copying, right? Because, again, they don't really want to make a mistake, right? That could mutate them. That could kill them. So the scary thing for us is the number of resistant plasmids out there. So these are easily transferred from one bacteria to another via conjugation. Although conjugation can involve stuff from the chromosome, a lot of times conjugation is transfer of the whole plasmid from one bacterial cell to another. And as I said, the bad news for us is there are a lot of plasmids out there that have genes on them that make the bacteria resistant to antibiotics. We refer to these plasmids as resistant plasmids, or R plasmids for short. So in the case of this picture here, this plasmid actually has resistance to 3 different antibiotics, right? So a penicillin derivative, tetracycline, and chloramphenicol. Notice the information on how to make the sex papillus is here too. So this plasmid is very easily transferred from one cell to another, because in addition, on this plasmid is how to make the sex papillus. So if they've got it, they can give it to all their friends via conjugation. So when cells have particular genetic abilities, they're referred to as F positive. And nowadays, too, you'll even see it'll say exactly what gene they have, that the normal wild type in nature bacteria doesn't normally have, right? So what does this bacteria have that others don't have? F minus infers that whatever gene we're looking at, they don't have. So again, if you have genetic information, you can usually make something, right? You can change your morphology, you can change your phenotype. So although conjugation can happen where chromosomal information is shared, we're going to keep it simplistic again too. And we're going to just talk about the movement of a plasmid from one bacteria to another. So conjugation via a plasmid transfer. So in this animation, you'll see that this guy has this plasmid, has the ability to make the sex papillus. Other bacteria have receptors for sex papillae on their surface. So this allows the joining to happen. So these two cells join up, and you can tell by the fact that this one has the plasmid and this one doesn't, right? This one's the donor and this one's the recipient. So they're going to join up. That creates a hollow tube between them. And he'll make a copy of his plasmid and send it over to his friend. Now they both have it and they can both share it. You could stick one E. coli in a test tube that has this ability. He will share it with all of his friends. It's about as bad as a sexually transmitted disease. Because again, this is bacterial sex, right? They live after they transfer this. Do you see that? Right? This is the only one of the gene transfers we talked about where the donor gets to live and keep donating. Right? He gets to go around, give it to all his girlfriends. Share and share alike. Oh, you laugh. They're having a symposium in March on sexually transmitted diseases, a microbiology talk, right? Guess when we get the announcement? This weekend. She's like, I didn't mean to send this out Valentine's Day weekend. I'm like, yeah, right. Chlamydia and all that kind of stuff. Like, really? So once one is negative, they can become positive. And the reverse can happen, right? They could lose the plasmid, right? They could kick it out of their cell and not utilize it. So conjugation is said only one where the donor actually gets to survive and continue to donate that information. So typically you're going to mate with someone opposite than you, right? So if you have genetic information, you're going to share it with someone who doesn't have it. They're not going to give it to one of the cells that already has it. But again, this depends on what you have, right? So typically, when we are talking, we'll actually specify what actual genetic information do they have or not have. But the reason why F is used as, what's the word I'm looking for? As an abbreviation, it stands for fertility. Again, you know, this having genetic information, this, you know, similarity to sex. Huh? F, just that designation of a cell, of being F positive, meaning they have this ability to produce the sex papillus and it has information that can be given to other cells. If you're negative, you don't have that. But you could acquire it, right? Once you acquire it, once the donor gives it to you, the recipient becomes F positive, fertility positive, and can share it with all others. Make sense? I think this is our last, nope. This one just goes through the steps. And then a picture. So again, they both become, as you can see here, F positive, right? And they can both go find other F negatives, make them F positive. Or, you know, chlamydia or gonorrhea positive. Okay, so this chart, the important take-home message here is to really help you remember and focus in on that the DNA is naked and it's all by itself in the environment. So if there are DNases in the environment, these are enzymes that destroy DNA, it would stop the ability of transformation. Where, remember, transduction, that DNA is where? That's traveling from one cell to another. Where is that DNA? It's inside the virus. So if there's DNases that can't get to it, the virus protein coat protects it. It's not going to be able to chew it up. It can't stop that process. The same with conjugation, whether it's a plasmid or transfer or part of a chromosome, those cells are joined with each other, right? The DNA is traveling through that protein tube. It's never in the environment. So if there's DNases in that environment, they're not going to destroy that DNA. That DNA is protected as it travels from one cell to another. So, genetic changes could result in a positive change for bacteria. How could this be? So, how could change be good? How could mutation be good? How could getting a plasmid be good? How could be picking up DNA from the environment or via a virus? How could that be good for a bacteria? What could they potentially get? Why do we talk about this? What are we afraid of them getting and sharing? Resistance to antibiotics. Right? Or anything that makes them better suited for the environment, right? Could be a positive change for them. And so because positive changes happen, these changes happen. Mutation happens. Transformation happens. Transduction happens. Conjugation happens. Right? Sharing this information can be beneficial. Obviously, gene transfers are much more beneficial than mutations, right? Because you know what you're getting, potentially. So that's it for genetics. Genetics is a bit of a monster for some of us, right? Which is why even though it's a later chapter in our segment of chapters, we covered it first. The next thing that we're going to go into is growth and control. And